Yeah, so our next talk is from Ted Kalbfleisch. I speak the name correctly?
You nailed it.
Yeah, from University of Kentucky.
Okay. I'm Ted Kalbfleisch from the University of Kentucky. I usually I'm in the Department of Veterinary Science, the Gluck Equine Research Center. Most of what I do is horse. I am here because I had breakfast with Peter Doris one day about 7 years ago, and he said, "Hey, I'm assembling some rats." Well, we had just built the ECCAB3 reference genome for the horse in my lab, and I thought, yeah, I'm game. So I joined him, and it's been a very fun ride. All right, so I'm going to talk to you about kind of sort of how we figure out whether or not we've done a good job, but the first part of the talk is going to be to give you some insights as to what's going on in the rest of the non-rat world. There we go. Okay, so for starters, this fellow George Box back in 1976, it's been attributed to him anyway, a statistician. All models are wrong, but some are useful. Okay. Okay, Tim Smith, a good buddy of mine, works at the USDA Meat Animal Research Center out there in Clay Center, Nebraska. 2002, made a similar comment. All assemblies are wrong, but some are useful. We're not going to get a perfect assembly, or at least not in the near future. But we can make them as complete and as contiguous as the technology allows. So that's what we're shooting for. Yeah. All right. So I'm going to take you way the heck back. The basis of genome assembly, since back in the days of the Human Genome Project, has more or less been the overlap assembly method, right? You take two sequences, you blast them, they align, and essentially they extend one another, right? So, you're going to do that with 30 million, 50 million, however many Sanger reads you've got, and you're going to build these long contiguous segments. But once you get to these repeat regions that we know to be nightmarish, like lines and signs and such, you get into those things, but you really have no idea where you're going to come out on the other side, right? You can't sequence through these repetitive sequences if there are 2,000 of them. That's a problem. Newer technologies such as long reads have made this attractable, have given us a tractable solution. But in the early days, these things were what beguiled us. So a timeline, right? So a lot of young faces here, so I don't know how many people know this history. Back in 1988, the Human Genome Project was begun, and in 2003, for the first time, the reference human genome was declared complete. We believe that for the last time, the human genome was declared complete just last year, but who knows? Right. So around 2005, between 2005 and 2007, this new technology, next generation sequencing, showed up. Oh, goodness. In 2003, it cost $2.3 billion to generate a genome sequence. $2.3 billion, all the king's horses, all the king's men. By 2007, we had that price down to $1 million. Right? If you want to drive the price of anything through the floor, give it to the biotech industry. 2008, RNA-seq burst onto the scene, and this was an era that was largely dominated by short-read sequencing. Everybody was doing short reads to find polymorphisms, to do RNA-seq and expression analysis, and it was a great time. A lot of really great science got done in here. 2018, Oxford Nanopore introduced ultra-long reads. So again, our big problem was contiguity. This looked like it could be a viable solution to it, although it was young and it was expensive. In 2019, PacBio announced Hi-Fi. If you can generate reads that are 15 kb long, that are accurate to 1 error in 1,000, that is transformative, and it was. PacBio Review got introduced, a couple of months ago, and now I know of several labs who are running that technology now. What isn't real clear here. Right in here, I don't know if anybody remembers this. There was like an X Prize. There was this thing, right? Someday we're going to see $1,000 to sequence a genome. When we do that, everything's going to change. We flew right by that, and nobody even cared, right? Yeah, I mean, we continue to make progress apace, and I'm still very excited for what's going on. Being able to build assemblies, and this is where the rest of the world is very much at a disadvantage from you guys. We had to, the rest of the world, horses, cattle, even people, we had to make pseudo haploid representations interpreting these diploid genomes, right? Copy number variations and haplotypes had to be inferred as opposed to directly observed. Why is this a big deal? I guess I don't need to tell you, but I did put it on the slide. Biology works with haplotypes. Alleles on the same haplotypes may have additive effects, so knowing where they are and which haplotype they're a part of is kind of important. Epigenetic imprinting on some genes results from different functions from maternal versus paternal copies of a gene. Knowing just what parent something was inherited from, also kind of important. The compromises, pseudohaploid representation. So if you've got like this maternal and this paternal copy here, that they kind of sort of look like this, they differ, you know, here, there's a line here, they've got these big repeats. There, you know, 4 of them here, 3 of them here, and you got all these single nucleotide variants. Well, when you've got some, you know, you sequence all of this and you try to figure out what the hell's going on here, and you put it all together, and it might look something like this, where you've got this region and this region that are actually the same thing. They'd be represented as nowadays they'd be represented as haplotypes, but they'd be thrown together into this same assembly, and it would be duplicated, and it would just be a mess. It was just unfortunate. Solution, so inbred animals, which is where you all are, and long read technology, really accurate long reads helped us out immeasurably, right? So if you've got a maternal and a paternal chromosome that are differing kind of like this, right? You're sequencing, and you've got these repeats. You know, where we kind of still are now, we're kind of resolving some of this, but you get to this and you'd recognize, all right, this belongs to one haplotype, this belongs to another. I'm going to build this one into the assembly, and I'm going to split this other one out as a haplotype. It's just going to be extra information in the assembly. I know I've got repeats, but I don't know how many, so I'm just going to leave them here. Okay. Now, rest of the world did something kind of fun, right? So again, these inbred strains seem to be the way to mitigate a lot of the problems that we were having with assembly. Tim Smith came up with this idea. Hey, wait a minute. We got these really accurate reads, right? So what if instead of trying to inbreed something, what if we really outbred them, right? What if we took 2 animals that were separated maybe by 4 million years of evolution? Let's put them together and see what happens. Okay. PacBio hi-fi data is so accurate. How accurate is it? It is so accurate that the PacBio reads that come off of the machine, if you know what the genotypes and the k-mers of the parents look like, you can actually take each of those reads and you can assign it to a parent of origin. So you can take the yak chromosomes and you can put them in one bin, and you can take this as a Highland bull here. You can take the Highland bull reads and you can put them into another, and you will then have 2 bins of what are haplotype resolved reads. What could be better? Spoiler alert, nothing could be better. This got itself assembled, and for, I don't know, some period of months, the most contiguous, most accurate, most complete genome out there for anything was a yak. Okay, so you know, again, the rest of us in the animal community, we've been taking advantage of this. The yak assembly I just told you about. These are some other projects that I've been involved in. Romanov and Dorper, those are both sheep. Again, the accuracy of these reads is good enough that even within species, you can do a pretty good job of separating these things. An Arabian, we crossed an Arabian sire with a Shire mare. A Shire, Arabian, really pretty horse. Shire is like a Clydesdale, only bigger. A where we're making great hay. And in fact, in the equine community, we're working to make a T-to-T assembly, is with a mule produced by a donkey and a thoroughbred. We're also working to sequence a zorse from a zebra sire and a quarter horse mare. And I was involved in a project that did a bison simmental assembly. Then I got to meet that Peter fellow. And after a couple of years, with the help of Melissa Laird Smith, we landed an NIH R01, and we put Kai to work assembling genomes. And this is the long list of what it is that we're working on. We've got the A3, the WKY, the B2, and the Brown Norway done, and a couple of the others are very near completion. The source of the material, you know these people, you know these rats far better than I do. Workflows in progress. We've generated about 35x. Now, if you were any other animal, right, that wasn't heavily inbred, you'd probably want around 50x of the hi-fi. But being really inbred, that 33 to 40 is going to suit you pretty well. Again, the SHRP, the WKY, and the SHRUTX, those have been submitted to NCBI. They've been submitted to Ensemble, as well as the ISO-seq data that we have generated for those animals. They are available for you to use and have been annotated by Ensemble. Okay, assembly metrics, size and contiguity. Kai talked a bit about this. The scaffold N50 and the scaffold N50 and the scaffold L50 were compelled to produce the numbers, but they're largely meaningless now, right? So if you tell me what your scaffold N50 is, I will tell you how long your chromosome 7 is, right? So I mean, it's useful. We've got to do it because essentially anymore, it's got to be the length of chromosome 7. If it's anything less than that, then you need to go back and you need to do it again. Okay, foreign contamination tool. Still, we have the problems of virus, bacteria, vector contamination that we have to worry about. NCBI has been good enough to put together a tool for us to look for it. And in the first 4 of these things that Peter's had a chance to look at, we can find no contamination. Okay, this is where the rubber meets the road. You take the lens through which we are looking at these assemblies is short read data. You take short read data from that animal and you map it to the assembly that it produced, right? Nearly all of it should map. And we're doing pretty well. In the short read data, we're exceeding 99% across the board. PacBio hi-fi data, and again, these are reads mapped to, so the A3 is mapped, the short reads for the A3 animal being mapped back to the A3 assembly, and so forth. And PacBio hi-fi, the mapping rates are even higher, which isn't terribly surprising, seeing as it was the hi-fi reads that built the assemblies. So, however good our genomes are, anybody who can afford to generate a short read or long read sequence data, they are going to have as complete a survey as the genome as is possible. Right. So when anybody generates that data, when they map it, we have to produce a reference that is going to be able to support nearly all of that data. And we've done a pretty good job here, I think. This is, again, when you're thinking about how complete something is, right? Does everybody know what a Kmer is? Some people? Okay, Kmer, it's just an arbitrary length of nucleotides. Right, so the Kmer size that we typically work with, 20 tumor, right? So it's PCR primers. You design a PCR primer 22 nucleotides long. It's going to be unique within the genome, right? So the search space, the Kmer space for a 20 tumors, like 1.7 trillion Kmers can be made up of 4 nucleotides. So essentially, what we do is we Kmerize the source data, we Kmerize the assembly, and we ask the question, are all of the Kmers that are in the source data also found in the assembly, and if so, how many times? And what we can see here, so this is a difficult, a bit of a difficult slide to explain. This peak out here at the center is essentially where every Kmer that is present on an autosomal chromosome ought to show up, right? And it does. And this little hump, wow, it's a touch screen. And then this little hump over here, that is where heterozygotes and hemizygotes would show up. So your sex chromosomes in a male, and any legitimate heterozygosity is going to contribute to that peak height. And this is data that was derived from long read data. And what we can see here is that nearly everything, it has the multiplicity that we want, and nearly everything is there. The size of that hump was a little bit troubling when I first saw it, but what our explanation for that is almost certainly reads from the Y chromosome that weren't properly incorporated. Y chromosomes are a nightmare, and all of these animals that we worked with were males. Okay, again, how, I could stand here and talk till I'm blue in the face about how great an assembly is, but if your gene is not represented in it, or if it's screwed up, then it's a terrible assembly, right? We absolutely 100% know that. So there's a new BUSCO benchmarking universal single copy orthologs tool out there that can be used to assess the completeness of a gene with respect to, we know that these proteins are present and conserved across nearly all species in a clade. Do we find all of those in your assembly? And for the most part, we're up in the neighborhood of 99.8% completion, so that's pretty good. But again, if your protein is in that 0.2 or 0.3, well, that's not so good. But hey, we are very much trying to get there. All right, I'm done. I am happy to take any questions.
Great.